Hello class, my name is Nicole Carnes and my presentation is on gene protein coupled receptors, also known as GPCRs. By the end of this presentation, students should be able to summarize the signal transduction pathways of gene protein coupled receptors, identify a gene protein transducer family and its associated second messenger, explain how GPCR mutations occur and their outcomes, such as diseases, and then discuss the importance of GPCRs in drug design and report the mechanism of action for alpha and beta androgenic receptors. Before we talk about GPCRs, we have to discuss the background information of cell communication. And in this section, we should be able to list the 3 steps of cell signaling and what are the roles and outcomes of signal transduction. Cells perform many tasks to maintain survival of the organism, and in order to do this, cells must communicate between each other and decipher messages signaled throughout the body. If a cell does not receive any signal, then that means it has led to cell death. There are 3 steps to cell signaling. Step 1 is reception, where the cell signaling molecule binds to a receptor protein on the cell membrane or inside the cell. Step 2 is transduction, also known as signal transduction. And this is when the binding of the molecule to the receptor leads to a conformational change, and the signal is converted into a form that the cell can respond to. Also, this activates the second messengers, which are used for GPCRs that are used to decode the signal. And then step 3 is that response to the signal. So understanding the basis of cell signaling serves as a building block for understanding gene GPCRs. So once a ligand is locked into a specific receptor, a sequence of biochemical reactions occurs in the cell's interior to convert the signal or stimulus across the membrane. This process is carried out by proteins, enzymes, and calcium ions that are linked through the second messengers, which include cAMP and IP3. And the second messengers relay the signal that result in a specific cellular response or even a change to the gene expression. So the roles of signal transduction, there are 3. So the first one is it enables extracellular molecules to affect cellular function without entering the intracellular environment. So the communication between cells is pretty distant, and this is accomplished by the binding of the agonist or the ligand to the receptor protein by stabling the structure in an active conformation. A second role is several different signals can affect one another by facilitating or inhibiting the activation of regulatory enzymic proteins by common or opposing metabolic pathways, and this yields a response to multiple stimuli. And then the third role is the activation of enzymes and the production of secondary messengers can be amplified and prolonged to produce a cellular response. And amplification is by a molecular cascade in which an initial signal is triggered by many intracellular responses leading to enhanced bonds. So it's a cascading. So it's small and then it gets bigger. Now that we discussed the roles of signal transduction, it's important to discuss the outcomes. So the first outcome is that there is a change in cell membrane polarity in the excitable tissues, such as the nerves and the muscles. There's also an activation of the cytosolic metabolic cascade, resulting in alterations of cellular morphology or function. And then it also leads to gene activation, where new proteins are synthesized, or there's also a modified cellular structure. Now that the basis of signal transduction is covered, now understanding how G protein coupled receptors function will be easier to uncover. Now, what is a G protein and what is its second messenger system? And what diseases are a result of a GPCR/G protein mutation? G protein coupled receptors are the largest family of surface receptors because of their various functions in the body. So another name for GPCR is the 7-pass transmembrane protein because of the structure of it, which is attached here on the right. So when you're looking at the structure of a GPCR, the transmembrane here contains amino acids at each end. So at the top here is the N-terminus, which is on the extracellular portion of the membrane. And then on the opposite end is the intracellular C-terminus. And then there are 7 membrane-spanning loops where 3 are intracellular and 3 are extracellular. Now, onto the function of GPCRs. So they are the most common receptor because it is involved in various physiological roles, such as sight, taste, smell, behavioral, mood regulation, and immune system regulation. There are more than 800 different GPCR that binds to various ligands, and there are multiple GPCRs for a single ligand that can activate a variety of transduction pathways and produce many cellular responses. So overall, a receptor is defined by its binding ligand, the signal is transduced, and the response that is given. Once the protein has gone through the 7 membranes of the GPCR, the signal is activated by a G protein, also known as the guanine nucleotide binding protein. There are 3 subunits to a G protein, which is alpha, beta, and gamma, which is seen here in pink on this diagram, and is also accompanied by 4 families. So there's GS, GI, GQ, and G12. But for this presentation, I'm only going to cover GS, GI, and Q. And all of that is transduced by a secondary messenger, AMP or IP3. So on the diagram on the right, you have the 7 transmembrane protein, or the GPCR, and the receptor activation leads to a change in the G protein where it triggers the release of a guanosine diphosphate, also known as GDP, and that is the smaller purple oval right next to the 3 subunits of the G protein, as you can see in steps 1 and 2. Then the alpha subunit is then replaced with a guanosine triphosphate, also known as GTP. That can be seen in step 3. Then the alpha subunit of GTP dissociates from the alpha beta units and binds to a particular target enzyme where there is either going to be an activation or an inhibition. And that is seen in step 5 and 6. Then the cycle continues where the alpha subunit catalyzes the dephosphorylated of GTP back to GDP, where the alpha subunit associates as alpha beta and the gene protein is in its inactive state, as seen in step 6 and going back to step 1. So this is a cycle of activation and inactivation between GDP and GTP. With the 4 G protein transducer families, there are second messenger systems that are associated with it, and I will discuss in detail about the second messenger system. So the GS transducer family, the top one, it stimulates adenylsilase activity and calcium channels, and that occurs when the GS protein stimulates the adenylsilase, which synthesizes ATP into cAMP, and this is occupied then by GTP. The cAMP binds to the kinase A, where the beta and gamma subunits dissociate from the alpha unit, where the signal is sent. And then you have the GI protein family, where its second messenger system, inhibits the adenylsilase activity and activates the potassium channels. So it is the exact opposite of the GS protein. And then the very last one is the GQ protein, where it stimulates the phosphorylipase C activity, which then hydrolyzes into DAG and IP3. And IP3 is a water-soluble molecule that diffuses into the cytoplasm, where it releases calcium from inside the cell, and then that calcium can bind to the protein calmodulin and protein kinase C, which stimulates other enzymes and proteins for phosphorylation. The signal is then terminated by the inactivation of IP3 by dephosphorylation, and that is where DAG is inactivated by phosphorylation to phosphatic acid and deacetylation to fatty acids. The final section of this presentation of GPCRs is the mutations that occur and their diseases, and also how GPCRs are used for drug design. So this section will cover what causes GPCR mutations and how are these receptors beneficial to drug design. GPCRs are responsible for keeping our body working properly, such as detecting hormones and growth factors, and then keeping our sensory functions working. However, there are mutations that occur with the receptors or the G protein itself, which do lead to many diseases that affect the physiological roles such as hormones and sensory functions in the body. There are a list of common failures that do occur. One being the failure to synthesize GPCRs or G proteins themselves, so there's misfolding. There's a failure to synthesize GPCRs or G proteins to reach the plasma membrane. There's a failure to bind or activate bind agonist. there's a failure of GPCRs to couple to or activate a G protein, and there's also a failure to G proteins to couple to its effector. Any mutation that does occur from a misfolding or it does not reach the plasma membrane, it does lead to a list of many rare diseases that are either a loss of function or a gain of function. And on the left-hand side are diseases that are caused by a loss of function with its receptor, disease, and its inheritance. And on the right-hand side is diseases that are caused by a gain of function of this mutation. The example that's given is a disease caused by a loss of function, and that is associated with the V2 vasopressin, which will be discussed in the next slide. An example of a loss of function is the diabetes insipidus, which is caused by a mutated vasopressin V2 receptor located on the AVPR gene. So this disease is caused by a decrease in the release of the antidiuretic hormone vasopressin, or a decrease in the response to the hormone, which causes an electrolyte imbalance, which then results in large amounts of urine excreted and consuming large volumes of water. So there is a fluid imbalance that occurs, which does not keep the body at homeostasis. So there are two types of this disease. There is central and nephrogenic. So central is caused by a lymphotic inflammation in the pituitary gland at the stalk and the posterior gland. Nephrogenic is the resistance of this antidiuretic hormone and is in the collection tubes of the thickening of the loop of Henle. So complications that do result from this disease are severe dehydration, constipation, vomiting, fever, inability, irritability, growth retardation, tachycardia, hypotension, fatigue, decreased body temperature, headaches, kidney and brain damage. And because the GPCR receptors and proteins regulate hormones, so if there is a mutation, this hormone is not released properly or does not have a proper response. GPCRs in drug design are excellent because they act on many signals, such as amine, peptides, proteins, hormones, proteinoids, and other sensory signals responsible for regulating physiological roles. And this helps medicinal chemists come up with drugs that will provide a therapeutic effect. So there are two types of androgenic receptors which respond to the amine signals from epinephrine and adrenaline. And this is the most common receptor. So there's agonist and antagonist, and they both have an alpha and beta subunit. And a given example is a beta-2 agonist, which is used for the treatment of asthma or chronic obstructive pulmonary disorders, also known as COPD. It involves G protein GS and the effector adenyl silase. When this drug is injected or consumed, it increases the cAMP concentration to activate protein kinase A and reduce calcium ions to prevent airway contractions, which it can be seen on the right-hand side here. So you have a beta-2 receptor, and you have the gene protein with its 3 subunits, and then it has the activation of adenosylase. Here it's going to make ATP, which converts to cAMP, which then inhibits calcium release, which then the smooth muscle is relaxed. An example of this is albuterol, which is a short-acting release beta-2 agonist, which smooths the smooth muscle for patients who are struggling with asthma or any kind of spasms. Build your learning. So there are 2 activities that could be implemented into the classroom to check a student's understanding. They can also be collaborated with a partner, or you can work on your own. So activity 1 is research a human disease resulting from a mutation in the G protein coupled receptor, and then identify if any medical treatment and its mechanism of action. Activity 2 is explain the mechanism of action of a drug that targets amine, peptides, protein hormones, proteinoids, and other sensory signals related to GPCRs, and list its mechanism of action. And here on the right is a table of the common androreceptor subtypes. So there's alpha and beta, and it has the different types of organs and tissues it targets and its response. You could use that to look up alpha and beta agonists and antagonists drug classes to help you with activity 2.